Locomotion and movement. Types of movement, that is amoeboid, ciliary, muscular. In amoeboid, it is macrophages, leukocytes, and amoeba, that is pseudopodia, right? And cyclo. Okay. Now the movement of our limbs, jaws and tongue, muscular movement and ciliary movement in trachea, and passage of ova through the female genital tract. And then muscle, that is flagellar movement. Then skeletal muscles, they are striated muscles due to the alternate arrangement of two proteins, actin and myosin. Then visceral muscles, these are like smooth muscles, non-striated muscles that taper at both ends, and these are involuntary muscles, help in transportation of food through digestive tract and gametes through the genital tract. So these are visceral muscles present in inner walls of hollow visceral organs like alimentary canal, reproductive tract. Okay, they are non-striated, no appearance, smooth in appearance, like cardiac muscles. These are like they show the branching, and they are striated, they involuntary, they are not under the control of our activities, that is central nervous system. Then that's it. Like muscle, the muscles have like sarcomere, it is a functional unit of the contractions, and like between the two I band, that is isotropic band, there is one elastic fiber, that is Z line, and there is A band, that is myosin. And important thing is that uh, the, myofibril, that is myofilament, is striated appearance is due to the distribution pattern of two different proteins, that is actin and myosin. I band isotropic band, A band anisotropic band. Okay, myosin. And that's it. There is like edge zone, it is the central part of the myosin, that is not overlapped by the actin filament. And then there is like A band, you see, and that's it. And going like structure of the contractile proteins, these are myosin and actin, like troponin, myosin, tropomyosin, F actin, like and then there is myosin. The myosin has like head, there is cross arm, there is LNM, HMM. Now actin binding sites and ATP binding sites present on head of the myosin, right? And then there is like one more thing, in the resting state, a subunit of troponin masks the active binding sites for myosin on the actin filaments. There is need of calcium. Now calcium comes from sarcoplasmic reticulum. The mechanism of muscle contraction. Now I'm going to read this paragraph right now. Muscle contraction is initiated by a single sent by the central nervous system CNS via motor neuron. A motor neuron along with the muscle fibers connected to it constitute a motor unit, right? The junction between a motor neuron and sarcolemma of the muscle fiber is called the neuromuscular junction or motor end plate. A neural signal reaching this junction releases a neurotransmitter, that is acetylcholine, which generates an action potential in the sarcolemma. This spreads through the muscle fiber and causes the release of calcium ions into the sarcoplasm. Now increase in calcium level leads to the binding of calcium with a subunit of troponin on the actin filaments, thereby remove the masking of the actin sites for myosin. Now, utilizing the energy from ATP hydrolysis, the myosin head now binds to the exposed active sites on actin to form a cross bridge. Now this pulls the attached actin filaments towards the center of the A bands. Now Z lines attached to these actins are pulled inwards, thereby causing a shortening of the sarcomere, that is contraction. Now you see, during the shortening of the muscle, that is contraction, I bands get reduced, A bands retain the length, and ATP binds and cross bridge is broken. So new ATP binds and cross bridge is open. So that's it. Like, and the breakage is repeated, causing further sliding. The process continues till the calcium ion pump back into the sarcoplasmic system, resulting in the masking of the actin filaments, right? Now the return of the Z lines back to their original position, that is relaxation. Now repeated activation of the muscles can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them. Okay. Now there is like myoglobin. It is a pigment that differentiates muscles into red muscles and white muscles. Okay. Now this is it. One more thing is, edge zone disappears during the muscle contraction and A band retains the length. That's it. Meromyosin, it is a monomeric protein in the myosin, and that's it.